Hello, and welcome to Knowledge Crusaders. Today's episode will be on genetics. Genetics was founded by Gregor Mendel, an Austrian monk who experimented on pea plants. He bred the plants together and then recorded the traits that they expressed through several generations in an attempt to understand inheritance, and in doing so, he laid the foundation for modern genetics. Every organism has its genetic information organized into chromosomes, and each chromosome is made up of many different genes. Each gene codes for one trait. For example, there is a gene coding for human eye color. Each gene has several different alleles associated with it, and these alleles code for a different expression of that trait. For example, both brown eyes and blue eyes have their own allele associated with the eye color trait. It is possible to predict the alleles that will be present in the offspring through the use of a Punnett square. This is a Punnett square. Each organism has two alleles for the same gene. So the parents would be here and here. As you can see, both of these parents have both a dominant brown eye allele and a recessive blue eye allele. The dominant allele will overpower the recessive allele. So in this instance, the brown allele overpowers the blue allele, and both of these parents have brown eyes, but they carry the blue eye allele. The four in the middle represent the offspring, and they allow us to predict a ratio for which the offspring can expect it to be born. There is one offspring in this set that has two capital B's, both coding for brown eyes. This is known as being homozygous dominant, as they have two dominant alleles. And 1/4 of the offspring can be expected to be homozygous dominant, since 1 out of the 4 listed here is homozygous dominant. 1/2 of the offspring can be expected to be heterozygous, since there are 2 offspring out of this 4 with a dominant brown allele and a recessive blue allele, and 1 out of 4 offspring can be expected to be homozygous recessive, as there's one organism here with 2 lowercase b alleles. It's important to note the difference between genotype and phenotype. Genotype is the alleles that the organism possesses, whereas phenotype is the trait that is expressed. For example, there are 3 different genotypes here: homozygous dominant, homozygous recessive, and heterozygous. But there are only 2 phenotypes, brown and blue. And in this example, 3/4 of the offspring would be expected to have the brown phenotype, as both the homozygous dominant and the heterozygous offspring would be brown-eyed, and they would possess the brown phenotype. Meanwhile, only 1/4 would have the blue-eye phenotype. This concept is simple enough, but sometimes things are inherited in a different way. There is codominance, and this can be seen in examples such as human blood type. In human blood type, there are 3 alleles. Lowercase I codes for the O allele, which is the recessive allele, and capital I with the superscript A codes for blood type A, and capital I with the superscript B codes for blood type B. The dominant A and B will always overpower the recessive O, but in the event that an organism has both the A and B alleles, they will have type AB blood, instead of A overpowering B, or vice versa. Sometimes you will hear the term incomplete dominance, and this is also just a form of codominance. There's also the example of sex linkage. There are certain chromosomes associated with sex. A female has 2 X chromosomes, while a male has 1 X and 1 Y chromosome. When they go through sexual reproduction, the female always passes on an X chromosome, as it is the only thing she has, whereas a male can pass on either an X or a Y chromosome. These chromosomes code for the differences between the sexes, but they also can code for different things. such as hemophilia for humans, which is located on the X chromosome. And therefore, it is easier for a male to have hemophilia versus a female, because the male only needs one hemophiliac allele, since he only has one X chromosome. Meanwhile, the female would need to inherit one allele from each parent. When the alleles of a certain organism are not known, a test cross can be performed, in which that organism is bred with another organism whose alleles are known, and then the offspring can be recorded, and based on their phenotypes, it is sometimes possible to figure out what alleles are present in the first organism, whose alleles are not known. There are certain ratios that are expected, as we see in the Punnett square, but sometimes when multiple genes are involved within the same trait, these frequencies can be disturbed, and this can sometimes be attributed to a condition called linkage, in which different genes associated with the same trait are located on the same chromosome, and when the organism is going through meiosis, the chromosomes will not be able to go through crossing over properly, when crossing over happens, those genes will both be transferred instead of them being separated, so they'll be inherited together, which can affect the final frequencies of alleles in the offspring. This has been Genetics. Thank you for your time, and come see us again on our next episode of Knowledge Crusaders.